Hello everyone, my name is Luisa, I'm a PhD student at Universidade de São Paulo and my advisor is John McNAMARA. My project is called Osmoregulation in temporo-spatially distinct populations of the freshwater shrimp Macrobrachium olfersii. This study was supported by CNPq and FAPESP. Osmoregulation is the mechanism responsible for maintaining osmotic homeostasis. These figures exemplify the relationship between internal and external environments and show ability for osmotic regulation, like in graph A, or osmoconformity, like in graph B. We can see shrimp as hyperregulator, green crab as partially hyperregulator, and mussel as osmoconformer. Most organisms have intermediate abilities. Osmoregulatory ability is the capacity to deal with physiological challenges and is directly associated with habitats. Macrobrachium olfersii is a diadromal shrimp species found in freshwater and estuarine environment and is distributed throughout coastal rivers in tropical and subtropical South America. It depends on coastal marine waters for larval development. My objective was to compare the osmoregulatory capacity in two temporally and spatially distinct populations from the northern coast of São Paulo State, Brazil. We compared data from the Guaicá River population in São Sebastião, generated by McNAMARA in 1987, with data from the Puruba River population in Ubatuba, generated in 2016 and 2017. Shrimps were collected by sieving the marginal vegetation and were transported to the laboratory and acclimatized in river water. They were then placed in the experimental salinities for 24 hours. Those conditions represent hyperosmotic challenge at 6 different salinities. Hemolymph osmolality was sampled to assess the effect of location, time, and salinity. We also compared the two distinct measurement methods used in 1987 by McNAMARA, freezing point depression, and in 2016 and 17 by myself, vapor pressure. For statistical purpose, we considered the mean osmolality of 2016 and 17, and location and time were considered as a covariable. Data were distributed normally, showing equality of variance. A 2A ANOVA was performed to evaluate the effects of location, time, and salinity, followed by SNK test. A linear regression analysis of the osmolalities was performed. A linear regression showed excellent agreement between the two types of measurement, demonstrating that different colligative properties of the hemolymph provide equivalent osmotic data. A 2A ANOVA showed that only salinity affects hemolymph osmolality. Location, time, in this case year, had no effect. These figures characterize osmoregulatory ability. We can see the relationship between hemolymph osmolality and external osmolality, that is, the environmental salinity. Hemolymph osmolality increased significantly in both the Guaicá and the Puruba River populations. Osmoregulatory abilities are very similar even after 30 years. In both figures, we can see that hemolymph osmolality increases significantly up to isosmoticity at around 600 milliosmoles, demonstrating strong hyperregulatory ability and then modest hyporegulatory ability. A similar osmotic gradient of about 400 milliosmoles is maintained against fresh water. We conclude that osmoregulatory capability in Macrobrachium olfersii is independent of long-term temporal and spatial variation. Macrobrachium olfersii is a strong hyperregulator and a modest hyporegulator. The two populations are probably polymicted owing to gene flow provided by common larval pool intermixed in coastal waters. Finally, resembling the Guaicá River 30 years later, Macrobrachium olfersii was no longer found, revealing local loss of biodiversity. The references, and I would like to thank my advisor, my university, and the financial support.